We will continue our discussion about the fatty acid oxidation pathway. In our previous videos, where we have discussed the experiments that has led to a conclusion that fatty acid oxidation plays an important role towards differentiation of embryonic stem cells towards definitive endoderms. So, in this video, we will cover what component of this fatty acid oxidation pathways plays a significant role or act as a bottleneck. And from the bioinformatic analysis of the sgRNA, the researchers of this paper has found out that it's the CP2 gene plays an important role in the fatty acid beta oxidation pathways. So how they have come to that conclusion? So from the bioinformatics, they find out that the CP2 gene RNA expressions or guide sgRNA expressions of the CP2 genes are increased significantly higher in the definitive endoderms other compared to its other genes such as CPT1AB, CPT1B, and CPT1A, CPT1B, and CPT1C. So those are not significantly upregulated compared to the CPT2. Though the CPT1B is upregulated, but its genes that are related to their other families are not upregulated. And let us first quickly understand where these genes are found and what are its role. These CPT1A, B, C, these genes are found on the outer membrane of the mitochondria, where its job is to convert acetyl CoA into acetaldine. And this CP2 gene is found in the inner membrane of the mitochondria, where it reconverts acetaldine to acetyl CoA, and thereafter the further fatty acid oxidation process occurs. Since in the outer layer of the mitochondria, out of these three genes only one is upregulated, whereas in the inner layer of the mitochondria or inner side of the mitochondria, this only gene that is responsible for reversal of this acetaldine to acetyl CoA is the CPT2 gene. So the researchers focus is more on the CPT2 gene as it's the only gene, and researchers have found it is significantly upregulated in the definitive endoderms. So the approach that they did is they prepared a cell lines in which they use the CRISPR Cas9 system to knock out the CPT2 genes, and they are able to generate two clones that is shown here. That through the genomic sequencing they can see that there is addition of one nucleic nucleic acid, and there is a deletion of 5 nucleic acids, and these things are been validated using Western blot also, where they do not see any CPT2 gene. And once validating that CPT2 genes are absent, they did the differential expression. They checked the differential expression of the genes in the CPT2 knockout compared to the wild type, and they can see that the CPT2 genes is significantly downregulated compared to the wild type, which is also they are expecting as CPT2 genes is knocked out in those cell lines. However, the genes that are related to the pluripotent stem cells markers such as SOX2 and ESR-RB are not significantly downregulated in this case, whereas in some case it is upregulated. Not ESR-RB, the SOX2 and POU5F1. So these genes markers are usually for this pluripotent stem cells, and they are not significantly downregulated. So this indicates that the CPT2 genes are still maintains the characteristics of the pluripotent stem cells, CPT2 knockout gene. However, the genes that are the markers for the definitive endoderms are significantly downregulated compared to the wild type, such as this SOX17, FOXA2 genes are significantly downregulated compared to the wild type. So when they differentiate these wild type cells and compare, and simultaneously they differentiate the CP2 knockout wild type cells to the definitive endoderms, they have find out that the gene markers for the definitive endoderms are significantly lower, around 40% lower compared to the wild type differentiations of the cells to the definitive endoderms. For example, this FOXA2 CXCR4 are results are shown in the panel 5. So around 40% of them are found to be downregulated, and when they check this using the flow cytometry, they can see the similar results such as this for this SOX17 and CXCR4. For the wild type, they can see around 59.8% populations has this SOX17 and CXCR4, whereas for the CPT2 and CPT2 one first clone and second clone, they can see the number of these gene markers are goes down. The number of these gene markers goes down. And this is the quantitative analysis where they were did a quantitative analysis of this flow cytometry data, and they can see the decrease in the CXCR4 and SOX17 populations in the CPT2 knockout clones. And these things are being validated using the immunofluorescence assay also, where they can see the more markers of these pluripotent stem cells such as OCT4 is high in amount compared to the wild type, whereas the markers for these definitive endoderms such as SOX17 are found to be lower compared to the wild type. And these are the merge image where we can see that lower amount of, where you can see the high presence of the pluripotent markers, whereas in the wild type there is no presence of pluripotent markers. This indicates that no or low presence of pluripotent markers. This indicates that CPT2 and CPT1, this indicates that CPT2 gene plays an important role towards fatty acid oxidation pathway that finally affect this differentiation of the embryonic stem cells towards these definitive endoderms. So I hope you now understand which component of this fatty acid oxidation pathway plays an important role towards differentiation of the embryonic stem cells to the definitive endoderm. And if you need to understand this experimental data or how the experimentals are performed in detail, and also need to access these supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.